Okay, so I think the number of participants joining is slowing down, so this is probably a great time for us to get started. Hello everyone, and very welcome back to the GHGA webinars, and Happy New Year from us. Today we will be covering a beginner's guide to data visualization. Before we get started, I would like to briefly explain what GHGA is. We are a genome consortium that works towards GDPR-compliant human omics data sharing in order to enable secondary use of clinical omics data in research, which we will hope will enhance biological discovery and replication of findings, and hopefully also translation of research insights into genomic medicine. And we involve a very diverse group of communities, which is a reason why we put these webinars on, because we're trying to get everyone to the table and discuss genomic research topics together. Before we get started, there's a couple of things I want to mention. First of all, this is kind of part of a little data visualization mini-series. So today we will be tackling a lot of concepts and ideas around data visualization. And then next month, Florian and I will be doing a more practical introduction. So we'll be using R and Python as coding languages to show you how to practically visualize some data. And if you're interested in other GHGA events, such as our lecture series, I highly recommend you check out ghga.de/events and sign up for whatever you're interested in. You can find all of our training resources like these webinars on our website under resources/training, and also on our YouTube channel. I will post these links and resources in the chat later on. This meeting will also be recorded, and you can find the recording on there hopefully in a couple of weeks' time. We also have a GitHub account where we will publish most of the slides for these talks, and I will be putting up these slides as soon as possible after this event. And our speakers today are Florian and myself, and I just quickly wanted to introduce Florian. He has a PhD in bioinformatics from the University of Freiburg. He works in outreach customer support and is a bioinformatic developer for Galaxy Europe. He has actually been on this webinar before and given talks on Galaxy as well. They're also on our YouTube channel. And he is currently also a bioinformatician and scientific workflow engineer for GHGA since April 2022.
Yeah, and I do the other side. Yeah, I want to introduce Julia. She has a PhD in molecular cell and development biology from the University of California in Santa Cruz. She is data scientist and data manager at EMBL and training coordinator for GHGA for quite some time now.
Thanks, Flo.
Okay, and then we start. Yeah, the beginner's guide to data visualization. Like you can imagine, data visualization is a very broad topic, and for the since we're in the GHGA, we have also a very broad audience. So it was quite a challenge to make this presentation and not running into a time problem, because you can imagine that depending which target you take, you can take hours to explain this. So it's really, this webinar is kind of the basics about data visualization. And first of all, it's important, I think you also intuitively, a lot of things you already grasp from it. Data visualization is, of course, a form of communication. It's a form of communication for us humans to make complex data and data in general more interpretable and more and easier for us to communicate. And there's of course a spectrum between information and misinformation. The sheer amount of misinformation grew over the years. I think that's important. That's why it's important to mention this here. When we're talking about information, it's really clearly stating the facts, how they are, And in the misinformation, we're talking about typical examples, which you quite often see. The best example is the percentages plots. So any type of data visualization with percentages, where if you catch a glimpse of it, it looks fine. But if you sum it up, then you will see that actually the sum of these subpopulations will not end in the total population. You see here right now already the example data which we decided to use. It was also a bit of challenging to find some nice example data. And it becomes a bit nerdy because it's about Pokemon. So we fall back into that type of into the data during this webinar. There's, however, another layer to think about when it comes to data visualization as a form of communication. From the scientific perspective, of course, we want to have it as objective as possible. So it's clearly just the facts. However, there came more and more into play a form of more subjective data visualization, which is called data humanism, which we will cover at the end of this webinar. And this is a form of data visualization that brings more the human emotions into play. This, of course, is not scientific at all anymore, but it can be used to make data visualizations more connective. So what are the key components to a good data visualization? Of course, if you talk to your colleagues or talk to your audience, the first thing is the data itself. And the second thing is which comes right next to it, is the visualization form. The second thing is your target audience, which plays a key role. This can range from a colleague or a person who's already familiar with the data and the data visualization, or somebody who's completely from a different planet and has no idea what you're talking about. So it's really important that you design your data visualizations with your target audience in mind. And the third thing is then also the location and context. So where do you present then also this data visualization? Is it more in a presentation? Is it more in a meeting? Or is it in a paper, for example, scientific paper? So with that, we come already to the first part, the data itself.
All right, and I will hop in here quickly from my perspective. Let's talk about some different types of data that we typically look at when we use biological data, for example. Flo, can you go to the next slide? So the most common types of data that we're using is either numerical or categorical. If we're talking biology, bioinformatics, numerical can be stuff like expression data, methylation data, invariant data. Categorical could be cell types, cell composition data, image recognition, transcription factor prediction. But we don't have to talk about just bioinformatics here. So one of my favorite data sets that I like to bring into these talks is either the Palmer penguins. So in there, the example would be something like the bill length or the bill depth. That would be a numerical data set to collect, or just the type of different penguins that live in a specific place, and that could be a different category. And going back to Flo's example with the Pokemon, an example there would be obviously the height or the size or the weight of a Pokemon and the different types: fire, water, grass, earth. Those could be categorical types of data. Okay, so moving on. There is a couple of characteristics in the data that we should keep in mind, that we want to have an eye on before we start visualizing data. A big one is data integrity, and that would be including accuracy, completeness, consistency, and validity. You also want to make sure that your data adheres to a certain quality standard. This can be very different depending on type of data you're working with. There's a security aspect to it. For example, if you're working with human data, Are you allowed to publish it? Are you allowed to process it on your computer? Stuff like that. And sometimes there's just the need to collect more data. Like, yeah. And so another question that is probably very good to ask yourself before you start visualizing data is, do you even have the correct type of data and the correct data set to answer the questions that you are trying to answer? And so since this isn't quite a data wrangling seminar, I just want to give you a couple of keywords here that you can look up in your own time and see if this is something that's relevant to you. If you are a bioinformatician or a biologist who's interested in these concepts, I can recommend the Good Statistical Practice webinar that Wolfgang Huber gave on this channel. He talks a lot about experimental design in bioinformatics, biology, and also his Modern Statistics for Modern Biology book. Now, just a couple of examples of things that you might want to look out for, one of them being messy data, which is sometimes caused by data not being formatted correctly. So one big example that makes its way through the biologist community is gene names. And gene names tend to be misunderstood by Excel for different dates. So I remember there being a gene that's MAR8, and if you paste that in an Excel column, it will automatically convert it to March 8th of some random year. And so if you end up processing biological data in Excel, you will introduce these errors. If you give that to someone else and they're trying to visualize it, suddenly you don't even have the same type of data in one column anymore. It's a big mess. So this is something that you want to look out for before you start processing and visualizing your data. And one principle that underlies this problem is making sure your data is fair. So it's findable, accessible, interoperable, and reproducible. And interoperable, for example, here would be really important if you're using Excel sheets and you're using them then again in a different software. You often introduce errors. And so again, since we don't have time to go into fair data and research data management, I highly encourage you to check out our resources on that topic. We have a whole course and we have multiple webinars on that topic. So the next thing you want to look out for is the completeness of your data, or if you have missing data. And here we're going back to Florian's Pokemon data, and you see all the different data columns that were collected. For example, one of them is height in meters, and a couple of other ones are the type categories. And so what you can see in gray is all the data that is present, and in black is some of the data that is missing. And so that's something you want to be aware of before you start visualizing your data and potentially misinform or come to wrong conclusions. And finally, one thing I want to mention is the different types of analyses that you might be conducting with data visualization. So we have descriptive, which is describing your data. What kind of measurements did you collect? What kind of observations did you make? Exploratory, this is exploring the different relationships between descriptive data points. Then you have inferential, which could be testing a hypothesis. Causal, finding the cause of an observation. And finally, prediction, to predict an outcome based on your data. And again, we won't be going into much detail about this right now, but I can recommend the statistics resources that I already mentioned. And this is something that we might get into more detail in the more practical webinar next month. And with that, I am handing over to Flo again.
Okay, so data we had now already. Now comes to the plots itself. And we are talking now about the individual things to the plots itself, which you should pay attention to. First thing is the axes. A common mistake to do is the axes. So here on the left, you have the penguin example of Julia, with the number of observed penguins on the y-axis, on the x-axis, the species. And one common mistake is to either, which you see here on the right, to split the axis, to stop it for a moment. There's on the upper left, upper right corner, and the lower right corner is to have a totally different origin where the y-axis starts. The problem with that, with the upper one is, and the lower one is, it gives a false perspective to the relation of the data. You can see clearly from the left to the right that, for example, from the shin strap, I hope I pronounce it correctly for the species, that in relation to the others, the distance is smaller or bigger. So it really gives false perspective to the data. Second thing where you can also influence the perspective to the data is the scaling. Here you have on the left side, again, with the penguin example, on the x-axis, the flipper length, on the y-axis, the body mass. And based on the scaling, which you see here on the right, so already also the size of the plot. So please take this into account. So if you go from rectangular to the square format, you influence the perspective of the data. It's not, the square is still fine here in this example, but of course, if you really, which you see in the lower right corner, change the scale of the x-axis massively, then everything is, I mean, that's not a very extreme case. Nobody would really do that. But I think it gives a clear example that it squishes, for example, the data together, and you cannot really, or you make false interpretation from it. Third thing is the color. One of my most liked topic for data visualizations. So we come to that to a moment. Of course, if you pay attention, there are associations to colors, which are technically very intuitive. For example, that gray obviously highlights unimportant data, which you can see here on the left and the right. Quite often you see this for log2 fold change plots, where you make non-significant data points gray, or here on the right, where you want to focus on a specific species. And so you highlight the specific species with red, which is clearly important. And on the left plot, for example, sorry, I also have a pointer. Here on the left, you can see that all the red points are basically significant data points. Another thing which you should keep in mind is, especially if you have a lot of categories, and we have this here with Pokemon. I did not actually know that Pokemon has so many types already. I'm from the 90s. I grew up with not so many. So if you have many categories, then of course you run into a color problem. So, sometimes you say actually you should not color to X categories, but it's sometimes unavoidable. So what is really important then is that you pick a color scheme which is more identifiable with more categories. So you can see here on the left and on the right, the right one is a bit more optimized. These color schemes you can find online, and they are also quite often supported by a lot of Python or R packages. So there's already options to find such color schemes which are visually more separable. And a good indication also for a good color scheme is if it's portrayable in grayscale. In the past, this was also a good recommendation to do anyway, because if you want to print out a paper or something, then sometimes you did it not in color, you did it in black and white, and then grayscale would help you to figure out if your plot is still interpretable, even though there is no color present anymore. It doesn't really matter. So this fact or this point doesn't really matter anymore, because we consume nowadays a lot of online media. Third point is that you have a consistent color scheme. It's also quite intuitive, but sometimes forgotten, to be honest. I see it in some papers here and there, that if you color your, for example, here your types, your water type in a certain color, that you keep this color throughout your whole plots. And a fourth point, which is, again, I put it in brackets, comes to print versus online. So keep in mind that print and online media use different color systems. look it up. So if it can happen, so if you use RGB, your RGB schema, that this doesn't, your plot totally looks different if it's printed out. So check that. And then, of course, so that's why it's one of my favorite topics, color impairment. This goes now already to a topic which we will pick up again, goes into inclusion. And color impairment happens more often than you think. And so I hope I will not now shock everybody with these plots. So this is a typical plot or visualization to figure out if you have a color impairment. On the left, it's technically, so if you have no impairment, so no visually impairment, and on the right is already adjusted to it. So for example, I have a red-green impairment, so I cannot clearly see what's written in here. It's not visible for me. I can see a 7 and 0. I already discussed it with Julia. It's supposedly not a 7 and 0, so I don't know. And on the right, it's adjusted. So I can here see now a 7 and 0. If it's really a 7 or 0, I hope so. So again, this happens more than you think. And also I see, since it's a personal problem, or not problem, it's a personal situation of mine, I can see quite often a lot of papers not taking this into account. Of course, this takes for you some time and resources to invest into making data visualizations more inclusive for this. However, there exist already some packages like Daltonize or Colorblinder in Python and R that helps you with that. So if you don't have a person in your team like me, where you can ask, can you see this or not? Then maybe use one of those tools which can convert your data visualization and make it more color impairment friendly. Then the next thing is, of course, resolution and title. That's, I think, also intuitive. On the left, I hope you can see it. It should be low resolution, on the right, high resolution. Yeah, if the plot is blurry, then it's not really nice to perceive. And B, if it's really badly, then you can also not really interpret anymore what your visualization should show you. I can hear sound. Sorry, can you mute please yourself? Yeah, so the resolution, of course, is quite often actually also a constraint in a lot of journals. So they will tell you which kind of DPI you should use. However, with presentations, this is obviously not the case, so pay attention to that. And the second thing is the title, or generally text in your data visualization. So really ask yourself a couple of times, is this really needed? Or if the plot itself, for example, here, attack versus defense for your Pokemons, if this is already, if that's already explains the plot itself. And if you already do it anyway, explain the plot in a presentation, then the title is, yeah, you can remove it. Third, or next thing is interactive data visualizations. That brings another layer to make data visualizations more approachable. Nowadays, there exist already a lot of tools which help you create or have a platform to do interactive data visualizations. One is, for example, the multi-locus view. That's data or graphical user interface for multi-ohm and also epigenetics data. There's also Galaxy, if you want to check it out, since I'm from Galaxy. There are a lot of tools available to do these interactive data visualizations. And if you basically more on the coding side, so you say, okay, graphical user interfaces are for me too restrictive because of you have to rely on what the framework what the functionality of the framework is. Then you can also use packages like Plotly in Python, which create interactive visualizations, which you can see like here. So to make it, so to explain, so you can then, for example, look at individual data points and see here that Chuckle, I don't know which Pokemon that is. I quite often only know the German words. Is a very outlier in defense, and that Hipini is a very bad Pokemon. Seems like to pick. Okay. Yes. Okay. That's then we come to the charts. Julia, should we already do a Q&A or should I continue?
I can't see any questions in Mentimeter right now or in the chat, so I would recommend you keep going.
Okay.
Thank you.
Then we come to the charts. There are a lot of data visualization types. And if we would cover now every possible type, we would fill probably not only one another seminar, but 2 or 3. So we focus on 10 here. These are kind of the most used ones. And I would anyway, if you do data science, then you will come across specific data plots anyway, which you use for certain types of data. And over time, you gain experience to use which kind of plot. And mentioning that, let's come to the first one, the bar chart. That's the kind of simplest one because it focuses on univariate, some univariate data. For our Pokemon example, would it be like, have the types, and you just look at the attack of those types. It's best to order the bar chart alphabetically or numerically, depending if your categories are in a certain order. And it's also best to flip the axes so that the labels are horizontally so that it's better to read. That's not a must. You can, of course, keep the categories on the x-axis and just then rotate your labels. It's also possible. But quite often, this flipping of the axis is a bit better, for readability. Then you have the line chart. This obviously focuses on non-cyclic data to show trends. In this example here, let's say we had the hypothesis to see if throughout the generations, Pokemon increased in strength or attack, let's say. So if you assume it's linearly, so you can use this chart to figure that out. So again, it's to identify trends, and you can also add uncertainties to the data visualization. Then there's another plot, the area graph. That's basically also a line chart, but with filled in area to point out the relative difference between the categories. it's again kind of a time series or non-cyclic data, and it might need additional annotation and explanation. You can clearly see this here in this example with so many categories. It's actually hard to see where which category has more. I think I did here now the number of Pokemons over generations. It's hard to see, so you might have to add certain locations, for example, here, to pinpoint the audience to the interesting sites. Then another very basic one is the scatter plot. This is, of course, used to describe relationships between two variables, here the attack and the defense between Pokemons. So we already saw that. And we already also mentioned that you can highlight areas in this plot if you use colors. I did it now here based on the categories, but of course, if you're interested in a specific category, you can then color it as red and the red gray. Then if you want to have another variable included or factor, then you can use a bubble chart. So here the size of the dots is then used, for example, here for the capture rate. So next to attack and defense, we have also the capture rate. So it adds another layer. And because it gets now more, because now you have more and more information in your chart, it might need additional labels. So where to look at, so to which Pokemon here, which Pokemon might be the most interesting one. Then you have the pie chart. Pie chart are used for comparisons of subpopulations. That's also a one where you can cheat quite often or make mistakes. So one mistake is, please check if the sum of the subpopulations end up to the total population. You can highlight also here certain subpopulations, so to just put it out or make a frame around it. And it's also best to order them based on the size. Then you have the heat map. The heat map is used for spatial or grid-based data. So the grid, we have here grid data for our Pokemon example. And I basically, that's the best example I could come up with. I basically calculated the fighting difference, so attack and defense pairwise, and just plotted here if the Pokemon or the first Pokemon would be stronger than the second Pokemon or vice versa. And obviously heat maps are used to identify dense regions or groups in your data. And here really important is to choose an intuitive color scheme. So like the hot to cold, so this one here from red to blue, or the other way around, to give an intuitive explanation to your data visualization. And then we are already almost at the end. So then we have histogram, box plot, and violin plot. So I grouped this here together because they all explain the distribution of the data. On the top, obviously, the histogram, and then below here, the box plot. The difference between histogram and box plot is here that histogram just focuses mainly on a single data set or the entire population, whereas with box plot you can obviously compare different categories here, like for the Pokemon, different types to each other. And with the box plot, you can then also see the variance and the outliers, whereas with the violin plot, which you can see here, that's this violin plot is a bit more complex, where it can be used to identify if you have multimodality in your distribution. So for example, here the poison type seems like it has two groups. So one with low attack and one with higher attack. maybe because the other one has more defense or less defense. Of course, you can add to the violin plot also information about outliers and things you see in the box plot, but this adds then more and more information. And then the question is if it's at some point too much. That's already it to the basic type of data visualization, or the basic type of. And then we come to kind of the principles. So these are a bit more now going into concepts for data visualization, and where you can maybe optimize, or with these concepts, you can optimize your data visualization. And one is everything everywhere all at once. Made a reference here to one of the latest movies. So the thing is that you should not put too much information into your data visualization so that the audience will end up in a state that they think nothing matters, and then they find it on a bagel. It's really that, especially these 3D graphs tend to do that it's too much. You cannot clearly see any trends in the data. And if you ever get a recommendation that you should work on your data visualization because it's too really then ask yourself, what is the key questions you really want to answer? And should it be really that detailed? Do you just want to give a basic overview so you can maybe remove some things in there? And what should your audience really perceive when it looks at it? And again, avoid 3D graphs. They tend to give you generally also a false perspective to your data. Second concept here is other forms of inclusion. I clearly want to say here, we now covered to include color impairments. There are obviously other forms or other things you can consider. However, this always takes more time, and you cannot include everybody. So it's up to you to decide how much you want to invest. But already some small things can help to make your data visualization inclusive. One thing, and this is also quite common, is this dyslexia. And to make a data visualization a bit more dyslexic friendly is to make it clear and concise. Again, mentioning it, that's not always possible. Sometimes a data visualization needs to be a bit more detailed, but then think about, can I create maybe an alternative way to consume the information I want to give, for example, to create a table? Or do I make it in some other way more dyslexic friendly by using different fonts, make more white spaces? I also shared here a link to the British Association of Dyslexia, and they have a pretty good guideline in how to make not only data visualizations, but also texts and articles more dyslexic friendly. Now we come to a concept which is called cognitive load. That can help you to figure out, is my data visualization the right, or is it in the correct format for my target audience? Cognitive load is a term which basically describes that each of us have some brain memory that we use to process the data visualization and store it in our memory. And this is, of course, from a light cognitive load to a heavy cognitive load spectrum. And this cognitive load can now be divided into 3 factors, which I will now describe now. And based on these 3 factors, you can then put your data visualization in a more light or heavy load category. What I mean with that is that you should not now get into a mode saying, okay, I see my data visualization is very complex, it has a very heavy load, and I should now change it to a light one. No, that's not the point. The point is that you figure out that your data visualization is on the more complex spectrum, and that also your target audience, so that it matches your target audience, basically. So any data visualization, light or heavy, is fine until, or with the intention, or with keeping in mind, when you keep in mind your target audience. So one factor is the intrinsic load. So the inherent complexity of your data, obviously. And so you have these these 4 things which contribute to the intrinsic load. One is the measurement itself. So if it's quantitative or qualitative. And imagine if it's more quantitative, so it's clear, has a clear unit to it, so your data. So like height is in centimeters. And this is easier to understand than a qualitative spectrum where you have like 4 levels, let's say 1 to 5. It's a bit harder, a bit more abstract to perceive. Then the knowability. So if your data is certain or uncertain, and with biological data, we actually always tend to be a bit more uncertain that your data is that particularly that this point of data has this particular value. Then specificity, meaning if your categories are more precise, like blood type, AB, or more ambiguous, saying like cell types. I mean, there are specific cell types, don't get me wrong, but also the cell states in between, they're a bit more ambiguous. So when do you decide what kind of cell types define cell states to define cell types, and so on. So this is a bit more ambiguous. And then you have relatability. So is your data more concrete? So for us, more relatable, like centimeters and body height, or is it more abstract, like the distance from here, planet Earth, to the next galaxy, which is in light years, which is hard for us to grasp. I mean, it's actually, if you see it, it's just a number, but you cannot really, it's harder to relate to it. Then you have the germane load. So this is the familiarity of the audience to the new information. I think that's also quite intuitive. So it's basically the connection. So when will your audience see the data visualization? Is it intentional, like you do now? So you in this presentation, or is it more coincidental? So they pass by, see the data visualization, and then go on. And then therefore, also the pace. So is it slow? Do you describe the data visualization step by step, or do you catch a glimpse and then you go on? Of course, then also the knowledge. Do we have an expert or this is more about the format or the framework this data visualization is embedded, and the confidence level with that. So it's like for you right now, so if you have no idea what this multi-locus viewer is, you less confident. And so you have to figure out first how to interact with this interactive data visualization platform. And then the last one is the extraneous load, which is the visualization or the form of visualization itself. There we have the chart type, where we have, it's either common or rare. Then we have the interpretation. So if it's, do you want to really from the audience to have an accurate interpretation? So this kind of fact they should get from it, or is it more like approximate is fine already? So like the data goes into a trend and they don't really have to know that it's, that's outcome A or B. Then composition, this we already mentioned, is it concise or detailed? And then the delivery, is it more explanatory or exploratory? Meaning, so are you explaining the data visualization step by step? So that's explanatory, or do you give your audience room to explore the data visualization themselves? And this basically you can put on this sheet here, and then you write it down, and then you figure out what the cognitive load of your data visualization is. Of course, it's some work to do, so you will not do it for all your data visualizations. So it's more a suggestion. So if you really unsure about your audience and how to make the data visualization better for your audience, then you can use this as a guideline to improve that. And then we come to the last topic. I'll take a zip for a moment. And that's data humanism, what I mentioned in the beginning. So that's the more subjective side of data visualization. And I want to stress out here clearly, so this is nothing you put into a scientific paper, because this the intention behind it is to make, to form for us humans a better connection to data and data visualization. So there is a person, Giorgia Lupi, who's quite famous for doing data humanism. To do data humanism. And she, I think she started with a project called Dear Data, where over 1 year she exchanged with a friend, Stephanie Posavec, I hope I pronounce her name correctly. Over 1 year, every week, they took a topic and for this topic collected personal data. So one of them was, for example, a week of complaints in general, grumpiness. I think everybody can relate to that. And they wrote, or they created this graph here. This is, by the way, one which I created because I wasn't sure if I can use the art or the data visualization on the website. And you can see here, they have here left and right. So the private complaints, the outward complaints, and each line represents one of the complaints. And the color represents the category. So if it's weather or work. And what they wanted to figure out is kind of the quote here on the left. Instead of using data just to become more efficient, we argue we can use data to become more human and to connect with ourselves and others at a deeper level. They really wanted to figure out if the data you already gathered, or the facts, if you can make it in a way that we humans can relate to it a bit better. And another project she did was called Bruises the Data We Don't See. I highly recommend to read the article. It's about another collaboration she did with a friend of hers. The mother had a daughter, or has a daughter of 3, at that time, 3 years, which developed I wrote it up here in the upper right corner, idiopathic thrombocytopenic purpura. I hope I pronounced it correctly, ITP. That's a disease, autoimmune disease, affecting the thrombocyte numbers in the body, which can lead to blooding and bruises, needs to some extent treatment. And Georgia Lupi wanted kind of to help her friend, because the friend obviously was in an emotionally what to say, intense situation. You can imagine if your child develops such a disease. And in order to help her, they collected the data, which was kind of the thrombocyte numbers, the platelet counts, which are these red spots. Each petal stands here now for a day. And they also collected other data like bruises, which was a smear here, or kind of the fear of, I think, the mother or the daughter, the fear level, so which was then highlighted by these lines. And they were grouped together, these petals. So these days, every time they went to the hospital. And so it is a timeline. Again, this is a representation. So I did this graphic here. So please look at the article to see the full data visualization. And so this work basically tried to answer two questions, which you can find here on the left in quotes again. Can a data visualization evoke empathy and activate us at an emotional level and not only at a cognitive one? And can looking at data visualization make you feel part of a story of someone's life? That was the question she wanted to answer. As I mentioned, it's highly subjective. It's not so scientific anymore. Of course, the data they collected, but not the data visualization itself. But it makes us perhaps more connected to the project itself. And so I want to kind of almost end this webinar with the last quote of her, but sometimes we completely forget the softer, more intimate, even fuzzy type of data. So it's really sometimes interesting to investigate if the data we collect, if there is another layer to it, which might also be interesting. That's at least one thing I interpret from that. With that, we come to an end. Again, it was a very basic webinar, but I hope you got some ideas about data and data visualization forms, which we talked about. You got an idea how to deal with your audience to figure this out. And I think what we did not really, so we briefly mentioned it with location and context. We can do it also with this cognitive load. And I think with that we are at the end, and I'm open, or we are open for questions.